Good morning to the guest of honor, renowned scientists from different educational institutions, audience, and my dear fellow mates. I, Yasha Nirati, presently pursuing Masters of Science from Institute of Bioinformatics and Applied Biotechnology, am delighted to host this inaugural session of Coronavirus webinar series. There have been many such series all over the world, but our main motto is to reach out to those people who do not have a good background of biology or life sciences, such as people in the field of social science, arts, economics, etc. We felt that there is an urgent need to come up with such a program so that people can grasp the basic knowledge behind the prevailing scenario due to coronavirus. There are lots of myths, confusions, and misunderstandings in the whole concept, so we try to pour it into a simplified way from the top experts in this field. I'm honored to welcome our esteemed guest, Dr. Vinod Scaria, Senior Scientist at Institute of Genomics and Integrative Biology, New Delhi. He is a pioneer in a precision medicine and clinical genomics in India. He is well known for scientists in sequencing the first human genome in India in 2009. He has established a large research network in the area of clinical genomics with the focus on the rare genetic diseases and pharmacogenomics. His initiatives have helped in uncovering the genetic basis of many genetic diseases in India. Recently, he has analyzed the phylogeny of various strains of coronavirus and has examined the time and place of origin of various clades. I would now hand over to Dr. Vinod, who will be talking to us about understanding the genomes of SARS-CoV-2 in India. Please, sir.
Thank you very much for inviting me here, and a very good morning to all of you. I hope the slides are visible. Um,
Yes,
Over
sir.
the next 30 minutes or so, what I'm going to talk to you is what we have learned so far from the many genomes of SARS-CoV-2 from India, and also draw your attention to the possibilities or rather more open questions which still remain unanswered. Before I talk and start my talk, let me first introduce what our lab works on generally. Um, we are not a virology lab, but we are a lab which calls ourselves doing genome informatics. Now, genome informatics essentially means that we look at genomes of organisms using computers. And you could address a variety of questions trying to look at genomes on computers. One of the major areas we work on is in the area of non-coding RNA or non-protein coding RNA. So these are regulatory RNA which regulates the rest of the genome. We also try to look at how the vertebrate genome is organized and what are the variabilities in the vertebrate genome, trying to understand how diseases could be caused or how the body plan morphology could be changed. We also work extensively with a large number of clinicians across the country on a variety of areas, mostly in the area of rare genetic diseases, where we co founded the Guardian Consortium almost 6 years ago. A very small part of my lab actually works in the area of pathogen genomes. We used to mostly work in the area of mycobacterium tuberculosis, but of course, very recently with the emergence of SARS-CoV-2 as a major pathogen, it just drew attention to look at the genomes of SARS-CoV-2. Of course, you could find more information about the institute where I come from and our lab at the website out there. Now, to start with, we'll need to first introduce the organism. And here the organism is a virus, a quite small virus called SARS-CoV-2. SARS for the severe acute respiratory syndrome, CoV for coronavirus, N which also stands for the novel. It's also called the CoV-2 because it is distinctly different from the CoV-1 or the first coronavirus epidemic that happened many years ago, at least a decade ago. The viral genome, as we know, is approximately around 30,000 base pairs. which is a quite large viral genome compared to most of the viral genomes that we typically talk about. The commoner ones, which have been extensively studied, for example, HIV, the hepatitis B, are typically in around 5,000 to 10,000 bases, and this is at least 3 times that size. And as we know, we also have read a lot of papers which suggest that uh, they belong to the genus of beta coronavirus and has been found previously in bats in China. Now, all that we know about genomes um, owes its origin to the first genome that was sequenced and deposited by Chinese researchers quite early in the epidemic. And this was deposited in February, uh, a couple of months after the epidemic broke up in China. In terms of nucleotide similarity, there are around 89% of nucleotides which are similar to the other coronaviruses, including the beta coronavirus subgenes. Now, what I want to draw your attention to is, unlike most large epidemics which happened in the past, there's significant differences and significant learning, at least, that we can take home from this particular epidemic. The first and the most important thing is that, unlike a lot of other epidemics previously, where people were grappling to find the identity of the organism that caused an epidemic, this epidemic was quite distinctly different, that we had not just the identity of the organism, but also the complete genome sequence of the organism. And the first genome, what we call as the WH human 1, or the reference that we use quite often, owes its origin to new approaches to not just identify the virus, but also be able to sequence complete viruses. Now, typically, when you talk about virology and identifying organisms in an epidemic, in olden times, it used to take a quite laborious procedure to isolate the virus, in many ways, culture, propagate the virus, and then be able to identify the organism. But here, you would see this, the technology has changed so rapidly that you could change the bodies, you could take the body sample, in this particular case, the bronchoalveolar lavage. You could do a RNA extraction, deplete the ribosomal RNA, and directly go back to sequencing. And this is what we call as shotgun sequencing. So you could take a body sample and you could process the body sample and sequence it directly. And then, of course, use the sequence information to reconstruct the virus and be able to know the identity of the virus. And that's the entire part of bioinformatics. This includes processes like base calling, includes QC, includes assembly, includes QC of the assembly, and of course, being able to put them in the phylogenetic cluster to be able to understand what is similar and what is dissimilar. Now, shotgun sequencing is possible today because of, one, that we have sequencers which can generate large amount of data, millions if not billions of base pairs of data in a single run. And of course, with that comes in the computational power to be able to put together the small fragments, typically 150 base pairs. So small fragments of reads back into assembling the complete organism. And this is what has really changed this particular epidemic, that we could take a sample from an individual without circumventing the entire traditional processes of microbiology, be able to sequence it. And more importantly, after you sequence it, use your bioinformatics capabilities to be able to assemble it back into the complete organism genome. If you think this is quite difficult, it is not. Uh, Very early on, at least in the month of March, we have put together this particular protocol, which students can easily follow. And of course, I believe you could take these links down so that you could go through these protocols. Many of these raw data sets are available in public domain, and you should also be able to assemble a genome with very minimal computational resources. You need an 8 GB RAM Linux system to be able to assemble these genomes. There is also a support group on Telegram, and you could avail this feature. Now, like all pathogens, whether it is viral or whether it is bacterial, the unique property about pathogens is that they evolve, and they evolve by mutations. The only different thing is that the speed of evolution, or the speed at which a particular organism accumulates mutation, is different for different organisms. So, for example, if you know that an individual could transmit a virus to another individual, and and then therefore form a chain, and this chain today we know for coronavirus is approximately around 5 to 10 days. So let's put an average of around 7 days. This particular virus can mutate approximately once in around 10 days. In other words, in every two transmissions, the virus undergoes or accumulates a new mutation. This has a lot of implications, and almost all that I'm going to talk in the next set of slides are going to be central around this very basic concept. And you will see this slide over and over again because this is essentially central to all that we know about virus and virus evolution, not just in coronavirus, but also in all the organisms and pathogens that we see. To give an example, you could see the first Wuhan Q1 genome, which was the first genome to be sequenced and deposited, was collected in December 22nd, 26th. And another genome that was sequenced from a sample collected in Jan 8, you would see that it has accumulated one additional mutation compared to the first genome that was sequenced. And moving forward, in February 27, the virus has accumulated multiple mutations, in this particular case, 4 mutations, which are different from the original strain out there. So this is how the virus mutates. As time progresses, the virus accumulates mutation, and of course, this is also an approach to identify and track back the virus, the origin, and the evolution. Today, we know a lot about the SARS-CoV-2 viruses, thanks to a lot of researchers. Despite being in the harshest conditions, they have ventured out into their labs, collected samples, sequenced the samples, and often uh, they have deposited the samples in public domain so that the rest of the researchers could access it and use it. And this is very different outcome of the particular epidemic that we are through. Unlike many other epidemics which were limited geographically and was limited to the number of genomes which are available, this particular epidemic, since it is also worldwide, has contributed to a very significantly large amount of genomes which are available today in public domain. To put numbers, we have close to around 60,000 genomes of SARS-CoV-2 in public domain, which is accessible to researchers like you and me. This also includes data which is deposited from individual labs. This includes data which are deposited by large consortia. For example, you could look at the COG or the COVID Genomics Consortium in the UK, which has deposited, I believe, close to around 26,000 genomes in public domain. And data sharing, therefore, is one of the distinct principles which is different from the previous epidemics of uh, any viral or pathogen origin. Now, of course, when you have a large number of genomes, there's also this problem of being able to access them, being able to search through them, and being able to slice the cake in the particular keywords that you want. And this is one of the resources that we have built quite early on. This is called the Genomepedia, where you could search the viruses which are available by the strain names, by the data source. For example, you could search for sources that come from Delhi. You could search for sources that come from Australia, so on and so forth. You could search by the country name. You could search by the lab which has sequenced it or the lab which has deposited. So there are multiple ways you can search and retrieve this information. I said before that the virus accumulates mutations, and when the virus accumulates mutations, they become a bit distinctly different from each other. And then, therefore, they could form lineages, and lineages are typically displayed on a phylogenetic tree. So, the lineages are otherwise, in scientific terms, also called as clades. So you could have clades of the same strain. So, for example, the SARS-CoV-2 is one strain, and you could have multiple small clades or lineages, each of them distinctly different from each other by a few genetic variants. And this is also going to be central to what I'm going to talk about in the next set of slides. Of course, you could also build phylogenetic trees and take in all the genomes that are available in public domain, whole or part of it. And there's an open protocol that's currently available, which is put together by one of my students, Bani. It's easily followed, and of course, you could download most of the resources which are also available in open source. Very early on in the epidemic, one of the resources that has come up is this very interesting resource called the next strain, which came out of the Trevor Bradford's lab. Apart from coronavirus, they have been tracking a lot of epidemics in the past, including, for example, the Zika virus, including the influenza viruses, and so forth. And this is how you could actually look at a phylogenetic tree and look at pathogen genome. So each dot in here is one pathogen genome. This is a limited representation of the 50,000 sequences which are available. And as you see here, they're distinctly classified into different lineages or different clusters, each of them distinctly different from each other by a few mutations. Now, if you take the global data, the lineages could be uh, delineated into 10 distinct lineages or 10 distinct clades. They're named uh, as A1A, A2, so on and so forth. The original isolates from China belong to the B cluster, the B lineage, and the A2A, which is a light blue dot that you see over there, is one of the largest cluster or the largest lineage which rapidly spread across the world. So, in many ways, the lineage could also point you to the origin of the virus or the location of the virus from which it came, and could also point to tracing the origin and introduction of the virus in a particular region or a particular community. Now, what is really important to mention here is that practically all the viruses across the world has been transmitted through people traveling on air. So in many ways, looking at a virus genome can also identify which individual from which part of the world actually brought in this infection. And from that point of time, the virus is going to mutate and evolve over the subsequent generations or the subsequent infection cycles. And then, therefore, you should be able to trace not just the origin of the virus in terms of where it come from, or what was the lineage of the virus to start with, and how did it evolve and emerge across subsequent infections in the population. And I'll come back to this again. This is possible because the virus mutates roughly approximately in 1 in around 10 days, and therefore it allows you to do what we call as molecular tracing or molecular ancestry tracing. Now, this has a lot of implications because it is not just individuals who have come into the country from abroad who have brought in the virus. That is what we call as an introduction. There are also people who traveled across the country from state to state. This could be people who have traveled not essentially on flights, but also on road. This could be people who have traveled on trains. This could be people who have walked all across the country. But nevertheless, they could all have, at least a small proportion of them could have carried the virus in terms of an infection and introduce them to a new population which was previously uninfected. And looking at the mutation, you can actually trace the origin and travel of the virus. And that's very essential because now you could be able to qualify the transmission across the world in terms of introductions. And now we also able to assess what clade or what lineage of the virus would emerge in a new area, provided we know what the introduction was. Now, coming back to the genomes from India, today we have close to around 1,400 odd SARS-CoV-2 genomes from India, deposited from a large number of laboratories, both medical as well as research institutions across the country. Today covers close to around 21 states and union territories, and uh, of significant mention is the Gujarat Biotechnology Research Center, otherwise known as the GBRC, in Gandhinagar, which has contributed a whopping large number of genomes from Gujarat, close to around 300 odd genomes from Gujarat, which means today we can understand all the epidemiology, all the introductions, and the evolution of the virus in one single state at very, very high resolutions. And this is really unmatched in the history of genome sequencing in the country. Even looking at it globally for a very limited population in Gujarat, this is probably one of the largest and the best data sets which span multiple time points across the epidemic of SARS-CoV-2. The obvious question that you would all ask is why sequence over and over again? Why sequence 50,000 times of the same strain of coronavirus? It was sequenced before. Yes, you need to sequence it because one, it allows you to understand the evolution of the virus. You can understand the origin or the introduction of the virus in terms of where did it come from and how is it evolving. And you can also trace community outbreaks if you can know understand where did this virus come from and from which contact did it come from. And of course, even more important is that this can allow you to evaluate diagnostics or in some cases even develop better diagnostics and also be able to understand how a potential vaccine or therapy would be efficacious or not. Now, if you look at the Indian coronavirus genomes, uh, we could cluster them into very distinct clusters, as you see here. 6 out of the 10 clusters which are globally prevalent, we have them. And apart from the six which are prevalent elsewhere or which have been documented in next strain, we have one additional cluster which you see here in purple. And this cluster is very distinct in the sense that it's a very large cluster only in India. And then therefore we call it as I or the A3I cluster. I, which says that it is a very distinct cluster in India. I'm not saying it's a unique cluster in India, but it's a very distinct cluster in India. And A3I to suggest that it diverged out of the A3 cluster of coronaviruses. Now, the A3 cluster of coronaviruses are present mostly in the Middle East, in the Persian basin. For example, if you say, look at Iran, so many of these green dots that you see here in the A3 cluster are essentially for people who came from Iran. Now, this cluster somehow is an offshoot of the A3I cluster. Doesn't necessarily mean that it evolved from one of these passengers who came from Iran, but nevertheless, it only says that it emerged at some point in time from the A3 cluster. Now, very likely that it didn't emerge in India because there were sequences of the A3 cluster which was found elsewhere in the world previous to the first introduction in India. So therefore, we surmise that this introduction and evolution had happened elsewhere, but nevertheless has become a very large cluster in the country. And this cluster today spreads a large number of populations across the country. And of course, this is not just a picture that I show. You could go back and browse this graph in this URL that's mentioned below. So this is an X-strain implementation of all the SARS-CoV-2 isolates from India. It's quite updated. You would find close to 1,000 odd genomes of high quality on this map today. I welcome you to all to go back and sort of browse this resource. There's a lot of more information that you can visualize on this resource. And like I said before, it is a mutations or the variants that distinctly define each of the clusters of the clades. Now, in some cases, the clusters of the clades could be defined by one. For example, if you look at the B cluster, the B clade, it is defined by just one mutation. It could be defined by multiple mutations. For example, if you look at the B1 or the A1A, they're defined by two mutations. And in some cases, the mutations could also be shared between clusters. Like, for example, if you look at the I or the A3A cluster, which is defined by four mutations, there is a fifth mutation which is also shared quite significantly in frequency with the A3 cluster. Now, so that's what I said, that you could also now look at lineages and sort of map how did this lineage emerge from a previous lineage. And this is exactly one of the reasons why we say that this particular cluster emerged from the A3 cluster, because it has one mutation which is quite prevalent and shared with the A3 cluster. Now, in many ways, these mutations are also mutually exclusive to each other, with exceptions, of course. For example, if you look at the B and the B4 cluster, the B and the B4 cluster shared by one mutation, but the B4 has an additional mutation. And the same is for A3I and the A3 cluster. And the same mutation is also, as you see here, shared by the A1A cluster, and so on and so forth. So, in other words, going forward in the future, we might not be able to require a large number of genomes, but now be able to just type these specific mutations which can classify a particular cluster of viruses. So, in other words, it might offer unique opportunity to build cheaper resources which can do large scale epidemiology on a very, very large number of samples to be able to understand differences between the clades in terms of, for example, looking at pathogenicity, looking at evolution, looking at infectivity, so on and so forth. Now, I said before that the virus mutates, and this mutation rate could be characterized because you have viruses and sequences of viruses spread across time points. So you can quantify over a specific period in time how much new mutations does the virus accumulate. Now, this mutation rate is exemplified by as sites per base pairs per year. And if you take all the isolates of coronavirus in India, uh, the mutation rate is approximately 1.48 into 10 to the power minus 3. So this roughly is 1.48 into 10 to the power minus 3 into 30,000, because you have 30,000 bases in the genome. So that's approximately 44 mutations per year. And that's approximately comes to around 3.7 mutations a month. And that's what I roughly said. It is approximately 7 to 10 mutations per week. Sorry, approximately one mutation every 7 to 10 days. Now, that's for all the viruses, and this pretty much concurs with what you observe globally. But of course, you could take individual clades of the virus and ask this question: do different clades mutate faster or slower than the average? For example, if you look at the A2A clade, which mutates at 1.42 into 10 to the power 3, 10 to the power minus 3, which is very similar to the average of the global mutation rate. You could, for example, look at the I clade, and you would see that this is much lower. This is 1.19 into 10 to the power minus 3. But of course, the A3 and the B4 are very small in number, so we might not really be really accurate in the estimation of the mutation rates. But nevertheless, to state that IA3I clade, which is distinct in India, finds a very small or much smaller mutation rate compared to the average mutation rate of all the clades put together. Now, the next obvious question you would ask is, what are the relative proportions of these clades across the country? And in fact, these clades would have very distinct proportions across samples from different parts of the country. For example, you could look at Delhi. Delhi has close to around 50% of the isolates belonging to the IA3I clade. You could go to, for example, Andhra Pradesh, right here. Andhra Pradesh has close to around 75% of the isolates belong to this particular cluster. You could now, for example, look at Gujarat, which predominantly belongs to the A2A cluster of genotypes. And of course, there are some states, for example, Madhya Pradesh, which also has predominantly the A2A cluster. Now, of course, you would find a lot of gaps in these regions across the country. And what we hope to is collaborate with a lot of laboratories across in these states from which, for example, there are no genomes or there are not much of genomes. This includes states like, for example, Kerala. This includes, for example, Andhra Pradesh, Telangana, uh, the northeastern states, Jammu and Kashmir, Himachal Pradesh, Orissa, so on and so forth, where there are limited representation of genomes, and then therefore we would like to uh, fill in these gaps by adding more genomes out there. Now, from the phylogenetic tree, apparently it seems that this particular clade, which is a IA3I clade, has been introduced once into the country and then spread across the different states, as signified by the relative divergence from a central point in the phylogenetic tree, as you see here. You could also look at temporal proportions of genome sequence from different parts of the country and ask this question: which clade of the virus is emerging, or which clade of the virus is devolving? And as you see here, the largest number of IA3I cluster came in the month of March and April, and then therefore has been slowly replaced, or is being slowly replaced by the A2A cluster of the virus. And this is very consistent across the different major states from where we have genomes. This analysis was done by Dr. Tej Saupati from the CSIR CCMB, who has extensively collaborated on this analysis. And of course, that opens up a new Pandora's box of questions. And the obvious questions that come to anybody's mind is, of course, if you have difference in the isolate or the genotypes of the isolates, is there a difference in the pathogenicity between the clades? And the pathogenicity is a broad term. This could be much specific in terms of there's a difference in infectivity, is there a difference in disease severity, is there a difference in the rate of immune response or the immune response or the outcomes for that matter. And of course, I stated before that I clade has a much lower mutation or substitution rate. And the obvious next question is, why is it having a much lower substitution rate? Is it because of very specific phylogeny or the topology of the phylogenetic tree, which makes us miscalculate this substitution rates? Because it is known that when you have a rapid emergence after a single point of introduction, you might artificially have a much smaller rate of substitution. This could also be because these are functional mutations, especially one of these mutation which characterizes this clade belongs to the RDRP gene, which is the RNA-dependent RNA polymerase, which is involved in the replication. So probably this could be a functional mutation which offers the virus a much high fidelity replication rate. And of course, you could access all these resources in terms of the genetic variants for all the isolates that have been sequenced so far in India. Today we have close to around 1500 odd mutations, and each of these mutations have been well annotated thanks to a lot of contributors, volunteers, research institutions, which have put the best efforts and contributed to this particular resource. This resource called Indicove is available on the web and it's publicly available. You could Google for it and you could find it. And for each of these variations, we have provided probably one of the most comprehensive set of annotations available anywhere on Earth. Um, so this is contributed by a large number of researchers who put together annotations from multiple different resources from their knowledge base, and this put together uh, essentially becomes a central resource, a central point where everybody can access the variants and their annotations. And of course, for students, there are very many open questions. Uh, Open questions are like, can genetic mutations change the efficacy of diagnostics? Can we understand the immune epitopes and how these mutations change the immune epitopes? And then can we evaluate vaccines in clinical trials, or even better, develop new vaccines? And can we understand how these viruses evolve and mutate, and also be able to define functional effects of the functional consequences of this particular mutations? Um, and one of these questions we tried addressing, and it's very pertinent in this particular scenario because practically all the gold standard tests that we use for diagnosis of SARS-CoV-2 is based on PCR, a very specific type of PCR called RT-PCR. Now, like all PCR reactions, the efficacy of diagnosis is based on how efficacious is the binding of the primer to the cognate target site. The primer is an oligonucleotide that the manufacturer has designed to identify and or bind to a very specific region in the virus genome and amplify a particular region. And once amplified, it is probed using another set of oligonucleotides, which is fluorescent label, and that's also dependent upon how efficient does a primer or probe bind to the cognate target site. So, in other words, theoretically, genetic mutations in any one of these sites, which are recognized by the primer or probe, could change the efficacy of the diagnostic reagent. Now, of course, this is more simple said than done. That's practically because the primer and probe information for majority of the diagnostic reagents available is not really cataloged elsewhere, especially even for large organizations, even national organizations, except, for example, the CDC or the Korea CDC or the Japanese CDC. For many of the national resources, the primer and the probe information is not readily available. So therefore, what we did was to go back and email a large number of companies who produce these reagents and ask the question, what could you share the primer and the probe information? And this was done by a large number of students from Bangalore. Of course, I have the names in the last slide, who put together this very large compendium of around 87 primers and the probes from different companies who willingly shared this information. And we asked a very simple question: for the primers and the probes, do we have mutations in the genome? And if they have mutations, how frequently are these mutations? So the plot that you see here is essentially the coronavirus genome. The y-axis is the variant frequency for variants, the 1500 odd variants in the genome. The ones which are marked red are essentially the ones which fall in either the primer or the probe recognition site. So put these numbers together, there are 123 SNVs and two unique indels which fall in at least one of the primer or probe site. There are 13 unique variants which have an allele frequency of more than 1% of the genomes sequenced out there. In other words, these variants could contribute to either false positives or false negatives in more cases for the diagnostic reagents. And as you see here, some of these allele frequencies as high as around 30%. In other words, very unlikely that the reagent that has a particular primer would be working in a country like India. Uh, Apart from this, of course, there are a lot of programs, projects, and data sets which are available in public domain. This is from our initiative on coronavirus genomics. I welcome you to go through this website, and if you have a lot of time to volunteer, please do volunteer. We also have put together a large number of resources to enable researchers, not just in India but also globally, to be looking at coronavirus genomes much more efficiently. This includes resources for phylogenetics. I did show one of the examples. This includes the genomic variant repository, which is Indico, which also I sort of mentioned, and we also have resources which can share genomic information in global standards. This is a genome beacon that we have built with the Dennis Boer group at Australia. And before I stop my talk, let me acknowledge a lot of people which made this possible. A lot of people who put in effort during the lockdown period at their homes to put together data sets, to put together an analysis. In many ways, many of them contributed wholeheartedly, including volunteers from Bangalore who contributed to putting in, for example, the primer of the probe information. And of course, there are a lot of students from the lab. This includes Bani, Mercy, Kavita, Mukta, Disha, Abhinav, Asta, Mohit, so and so on. Fantastic collaborators, Sridhar for most of the NGS that we do, and fantastic collaboration from CSS CCMB, which is a sister lab, which also has extensively invested in looking at coronavirus genomes. Um, and we are deeply indebted to a lot of researchers from across the country who have put together the genomes from the respective states and made it available in the public resources, either the GenBank or the GISAID datasets. And of course, this is all possible with funding, and funding in almost all senses came from CSIR. I think I'll stop there and take questions. But before that, let me also use this opportunity to invite you to participate in this social endeavor to understand COVID-19 disease. So this is a web app which allows you to track the progress of COVID infection. So this is essentially for people who are not yet infected. You could enroll yourself to this particular program, and on a regular basis, we would get back to you asking very specific questions, trying to understand what are the risk factors, what are the comorbidities, and also assess what are the outcomes of infection. And this is under the assumption that a significant large number of people, in our assumption, close to around 20 to 30% of individuals in India would get infected with coronavirus at some point of time. And uh, this data and a crowdsourced approach would allow us to learn a lot more in not just understanding the pandemic, but also in many instances to be able to be prepared to the next pandemic. I think I'll stop there and take questions. Thank you.
Thank you so much, sir, for such an informative talk. Your explanation has enlightened us with various aspects of today's topic. We will surely participate in your initiative too. I'm pretty sure people still have lot many questions and queries in their mind, which we will surely entertain after a short discussion. Audiences are requested to show up in the comment section in case of any query related to the discussing topic. So, sir, we'll start with a very basic thing, and it's my personal question as well. That since we know that the coronavirus has been there for a very long time, but the pandemic has appeared last year. So, what do you think are the factors which has led to this situation? What are the basic factors which has created such huge
Yeah, so
difficult
I
situation?
would answer. Yeah, so I would answer this in a very general perspective. If you look at the very recent epidemics or outbreaks of large mortality across the world, this could be, for example, the present coronavirus. This could be the previous SARS infection. This could be, for example, the Ebola virus. This could be the Nipah virus, and so on and so forth. Generally, um, what we see is that these are viruses which has emerged. These viruses have always been uh, there in the animal host, but these are viruses which has also jumped from an animal host to the human host, and this could be of multiple reasons. The first and the foremost reason is that humans being in contact with animals have increased. Um, I mean, uh, across the globe, we have forests rapidly being depleted, and this has contributed to the animals coming into close contact with humans. The second is, of course, as a virus evolves and virus mutates, at some point of time, it evolves to such a level that it can recognize the cognate human receptors that you can now start jumping the species. Now, but nevertheless, having said that, the virus, despite having a mutation that can jump the species, will not jump the species until you have direct contact with the particular species. For example, if you look at bats, the bats would have coronaviruses which can identify the human receptors. But that doesn't mean that it will always emerge as an epidemic until the bats come into contact with humans. So it points to a much larger question about the global climate change, and many of the pandemics that we see and that we have seen in the past, and probably that we will see in the future, would be contributed to a large extent by by the global changes that are happening in many ways, humans invading into the animal territory.
Okay, sir. So I also had this confusion in my mind that there are speculation all over the world that it has been created or developed in the lab, and one of the Nobel laureate has also supported this notion. Luc Montagnier is one of the Nobel laureate who has supported this notion that it has been intentionally developed in the lab. What do you think? Is there any support to this notion? What are
So the
your
present genome
viewpoints to that?
analysis doesn't prove or disprove any one of these hypotheses. So I believe that they should still just remain as a hypothesis without validation until the
Okay, sir.
validations come in. Yeah. So at the present genomic data doesn't either prove or disprove this altogether.
Yes, because they have found some genetic elements of HIV virus and Plasmodium falciparum. So they're saying that presence of such genetic elements which are not related to this strain of virus is cannot happen naturally that too this early. So it can be that it has been developed in the lab. So that is why I had this confusion. Do you
Yeah. So that's
think
what I
that
said. Given the genomic data that is available so far, um, it is not sufficient to prove or disprove it equivocally. So because when somebody needs to make that statement that it was developed in a lab or it was not developed in a lab, you need to
hmm,
have quite conclusive, very strong evidence to be able to support that. And at this moment, we are very far away from that kind of an evidence which can prove, or for that matter, even disprove that observation.
So, as we know that the virus binds to the receptor. So, till now, as per the recent researches, there are cluster of amino acids at the protein spike, which binds to the receptor of the cell. It's like a key to the cellular lock. So, we say that in case of coronavirus, it is the ACE2 cellular receptor that is involved. So, do you think is there any other receptor involved to help the virus to spread and infect the cell?
Yeah, so apart from the AC2 receptor, there are also other receptors for the virus. This includes the TMPRSS receptors and many others. But having said that, ACE2 is supposed to be the major receptor for the recognition of the virus. Uh, there could be many other more receptors still yet to be characterized. But yeah, having said that, there's not much of research that has gone into recognizing the whole plethora of receptors in humans. There could be many more receptors, probably of much smaller affinities, and then therefore could also have a lot of implications in the disease pathogenesis. But yes, we don't have too much of evidence to suggest that.
So
Most of the
basically,
evidence is centered, yeah, most of the evidence is centered around the ACE2 receptors.
okay, so sir, if there is a probability of having some other receptors also, so we cannot say or we cannot validate this fact that there is a complete lock and key mechanism between the cell receptor and the virus spike protein, because we cannot say that they are highly specific to each other, since we have some idea that there can be any other receptor other than ACE2.
Yeah, so to answer that rightly, so the virus didn't evolve seeing the ACE2 receptor, right? I mean, the
Okay.
virus was in bats and it was always looking at the bat receptors. But at some point of time, just by accumulating mutations, like I said, every 10 days or so, the virus accumulates
Hmm.
mutations. And just by accumulating mutations, you would make proteins which can now start recognizing proteins in humans, right? So this
Okay.
could be AC2 receptor in this particular case, but this could be some many other receptors in many other cases of other viruses. So it is just that by chance that you evolve to recognize a particular receptor, and using that affinity that is comparable to the cognate receptor in the cognate host, you can start to jump the species, right? So
Okay,
it is
sir.
a random event, and it's a matter of evolution. You can just think of it in terms of the possibility. If the virus creates 44 new mutations every year, and just think the virus being in bats for the last 10 years, that is what,
Exactly.
400 odd mutations, and it's quite large for a 30,000 base pair virus,
Exactly,
right?
sir. And
So
also, there are 20 amino acids, and to have that exact permutation and combination of amino acids on the spike protein, it's very difficult. It cannot happen
yeah,
in a
so
one go.
no, it has not happened at one single go. It has probably
Yes,
happened, and it has probably been there for quite some time, right?
yes,
And
yes, yes,
having
sir.
said that, there is also a very large possibility that many more viruses, not just coronaviruses, many more viruses in animal kingdom, which could at some point of time recognize host receptors in humans,
Okay,
right?
okay, sir.
They're probably been there, they probably are there, and probably will be there in the future.
Yes, sir. So, while talking about the cell receptors, we know that uh, cell receptor is present in most of the cells in the body. But when we transfuse the blood from the donor to the recipient, we see that the virus is not transmitted. How is this possible? Like, there have been some evidences where there has been blood transfusion from the infected donor to a healthy receptor, but there has not
I,
been any positive test.
frankly speaking, I have not followed the literature on this. So
Okay, sir. Oh, it's okay. It's okay, sir. It's completely okay. So, as you have discussed in your presentation, that the virus is mutating at a high rate in approximately at 7 days. Sir, so is there any notable mutation in the Indian strain of the coronavirus, which is only prominent, only found in Indian strain?
so that's what I said. So we cannot call it an Indian strain because there is only one strain of coronavirus, that is SARS-CoV-2. These are lineages of the same strain of
Okay, yes,
virus.
sir.
So it's not
Hmm.
very special species or a strain of virus. It's the same strain, but uh, bit different from all the other viruses by four mutations, right? Of this four mutation, one of these mutation, as I stated, is in the RDRP protein, which is the RNA-dependent RNA polymerase. And these four mutations are distinct from the other clades or other lineages of virus. It doesn't
Okay,
mean
sir.
that it is. So that's what I very specifically stated, that this is not exclusively found in India. It has
Hmm.
emerged elsewhere, has come into India, and are also present elsewhere. But anywhere else, it has never been in such a large number, as large proportion.
Okay, sir. So, is the virus at its complete potency, or it has the tendency to even spread more and mutate more than what it is now?
So the virus is mutating and it will keep mutating.
Okay.
Now, what will constitute to increase pathogenicity or decrease
Yes.
pathogenicity? Frankly speaking,
Mm.
we don't know,
Okay.
right? But having said that, the virus is mutating and will keep mutating.
Okay, sir. And sir, are these mutations only at the genome level, or some of the, of course, protein level mutations have also been there? So, is there any evidence that a particular region of the genome, when it's mutating, then only there is a mutation in the protein? Is there any such thing?
So obviously, so if you have, so when you call mutations, these are essentially genetic variants. These are nucleotide changes. If the
Yes.
nucleotide changes fall into a protein coding region of the virus, and
Hmm.
it changes the codon, when what we call as a non-synonymous mutation,
Hmm.
then all the non-synonymous mutations will cause a change in the amino acid in the virus,
Yes, sir.
right?
Yes,
So
sir.
there are non-synonymous mutations for sure, including the one of the mutations that we found in the eye or the A3i clade, and it can change the amino acid, and then therefore can change the protein sequence. And if the protein sequence is changed in a very functional region or a very important region, then you could also expect the functionality of that protein to change. So there
So,
are protein coding changes also.
okay, so the mutation that you're saying, there is a mutation approximately at 7 days of the development of the virus. So, I wanted to know that that mutation is at the genome level or it is at
It's
the
at the
protein
genome
level?
level. It's
Because,
at the genome level. It's at the
okay,
genome
it
level.
is at the genome level. Okay,
Yeah,
okay.
there are
Maybe sir,
substitutions at
because
the genome level.
yeah, because substitution in genome level might not always lead to the mutation in the
Absolutely,
protein.
absolutely,
So that is
absolutely.
why I asked these mutations are considered for genome level or for the
At
protein
the
level.
genome level. At the genome level.
Okay, sir. Okay, sir. So do you think at some point of time, the way the virus is mutating and is evolving, it would lead to the failure of the diagnostic kit?
Uh, no, this is a very interesting question because yes, answer is yes and no.
Hmm.
Yes, if the diagnostic reagent that you're using is disrupted by the mutation. Like I
Okay, sir.
stated before, there are a lot of diagnostic primers and probes, especially in the N gene, which also have very high frequency across the Indian isolates of SARS-CoV-2. At least one of these cases has around 30% of the strains having a mutation. Now, yeah, briefly speaking, if you understand thermodynamics, you will easily understand this principle that you have a primer or probe which is designed to bind 100% to the cognate site. So if you have a particular genetic mutation, that will cause a mismatch, and this mismatch will change the delta G for binding, the free
Yeah.
energy for binding, and this
Yes,
will
sir.
also change the Tm for the reaction, right?
Hmm.
Which will make the reaction much lesser efficient. So yes, mutations could change, and this change is dependent upon the thermodynamic properties and also depend upon the position of the mutation on the cognate target site. For example, if you have at the 3' end of the primer binding site, then very likely that the primer is not going to work, right?
Yeah.
Because it's a large base pair, and if it doesn't
Hmm.
match, then you're not going to extend, right?
Yes,
So
sir.
that necessarily
Sure.
means that you are going to get a false negative report for that particular genetic mutation. So it's a mix of both thermodynamics as well as the position of the mutation on the reagents that you build. Yes, so for some reagents, yes, at some point of time you will have the reagents not work as expected, right? So what you would need to do is to go back and address this question and try to build new primers or probes into regions which are very unlikely to mutate in the Indian isolates.
Okay, so we need to keep a very deep track on the rate of mutation, the changes in the genome sequence,
That's
right?
that's right. That's right.
Yeah, yeah. So, sir, these days we are using some anti-malarial drug like hydroxychloroquine and antiviral drug. These are giving some positive outcomes, but do you think we can totally rely on this, or should we come up with some drug which could be be some high percentage of effectivity, should be shown towards the inhibition of the viral growth?
Yeah, so there are a lot of drugs in investigation. The last count that we had was around 141 molecules, which are in some kind of investigation. Um, the point that you need to keep in mind is that typically drugs and the utility are assessed through randomized controlled trials, and randomized controlled trials done in different populations and showing consistent results is what typically will suggest as a particular drug to be used in a particular infection. Now, having said that, none of these drugs or none of these molecules have gone through very rigorous multi-center randomized controlled trials to be able to conclusively say that this is the drug that we need to use in coronavirus. At this moment, we do not have a drug which which we can use, which can cure 100% of coronavirus cases. So, having said that, yes, it is still an investigation, and probably we should wait for more data to come. And of course, these are not very simple experiments or very simple studies that need to come, because you need to control for a lot of co-occurring factors which can skew the observations. So, yeah, going forward, we expect to see more data on more molecules with randomized controlled trials, and I think that is what's really, really important.
Okay, sir. So, I've also read that this anti-malarial drug is an immunosuppressant also. So then, how this plasma therapy is working? Because in the plasma therapy, we are using convalescent plasma. That means the plasma is taken from the already diseased individual who has recovered, and then this plasma is given to the patients who are showing mild or are having some different health issues also, to treat the disease, and it has given some good results. So, sir, if it is suppressing the immune system, then how are we getting the antibodies for the plasma therapy then?
Yeah, so plasma therapy is also an investigational drug. That means it is still under investigation. It is still not approved for treatment.
Hmm.
Uh, Initial studies, so the plasma therapy is under the hypothesis, and very importantly, I qualified as hypothesis, that you have antibodies, and many of these antibodies could be neutralizing to the virus, and then therefore, if you transfer the plasma from one individual to another individual, you transfer the neutralizing antibodies, and then therefore the neutralizing antibodies act upon the individual, right? Now, having said
Okay.
that, plasma therapy is still an investigational drug, and at least the very recent reports do not show such a very large effect in terms of treating COVID patients. In India, at least, there are multiple centers who are doing the trials, and we are still awaiting the results for the trial.
Okay, sir. Okay, so people are trying over developing a vaccine, and I recently have read some successful testing of vaccines to some stage. So, sir, where do you think it must be failing before, or still it has not been completely successful? So, where do you think in what stage it's lagging behind?
See, we are probably today in July for an epidemic that started in December, right? So I think um, we should also moderate our expectations on a new drug or a new therapy, right?
Yeah.
Um, so if you look at the history of the vaccines which have been developed before, um, it takes a lot of effort, and it takes a lot of good science to be done before you can really put out a vaccine, right? And
Okay.
at least in this particular case, there is a lot of more science to be done to really understand how even the virus is causing the disease, right?
Okay.
The great example to this is, for example, the fact that many of the people who die out of COVID is out of a hyper-inflammatory response, right? That means
Okay.
that your body is eliciting a more than natural immune response to the
Immune
virus,
response.
and this extra immune response is what is going to damage the tissues and damage the host.
Uh-huh.
Now, until we really understand how this virus elicits that kind of a hyperinflammatory response, you're not going to understand how I mean, what arm of the immune system that you need to really target to build a good vaccine. Is it the humoral response arm that you need to target, or is
Yeah.
it the T cell response that you need to target? We are still, uh, I think, a bit far away or from the reality. Yes, having said that, there are of course vaccines, and many of the vaccines are in clinical trials.
Yeah.
But I think what's really more important, especially for students, is to also go back and understand the basics of immunology, and trying to ask very important questions in uh, what are the properties of a good vaccine, right? And you will
Yes.
really realize that there's a depth of information on what constitutes a good vaccine, right? We
Yes,
really do not
exactly,
know.
sir. Huh. Because also the strain is new strains are coming, the virus is evolving. So it depends on what the basis we are developing the vaccine. Because if the strain is changing with the period of time, we have to develop a vaccine which is effective to most of the strains at least.
That's
Otherwise,
right.
we cannot. I don't think it's practically possible to develop a vaccine for a particular strain for a particular geographical area and then administer to people and help them to come out of the situation.
Yeah, so my quick answer to that question is, at this moment we don't have a vaccine, right?
Yeah.
And until we have a vaccine, we would not know how genetic variations in the virus is going to impact that vaccine,
Yes,
right?
sir.
So
Yes, sir.
it's so at this moment, talking about variants which could impact vaccine is like putting the cart before the horse, right? you have to first
Okay.
get an you have to first get a vaccine, and that vaccine has to work. And if given a vaccine that works, then we can actually go back and ask this question, that what makes it not work, right?
Yeah. Yes, sir. Yes, sir. So can we not do this that we can develop a kind of an inhibitor which could inhibit the glycoprotein of the virus, which so that it can help to inhibit the spread of different viruses and not only the coronavirus. Other viruses are also not able to spread throughout the body just because of that inhibitor, which inhibits the glycoprotein of the virus. Can we do this? Can we
See,
develop such
theoretically,
flexible glycoprotein?
yes. Theoretically speaking, answer is probably no. And one thing that you need to keep in mind is that almost all viruses are not completely external, because essentially when you look at coronavirus, or for that matter any viruses, they're all obligate parasites. That necessarily means that
Yeah.
they are carrying the very minimal genetic information that they need to replicate and pass on,
Hmm.
right? And they obligately use the host machinery for all their activities.
Yes, sir.
So when you say glycosylation of
Absolutely.
proteins, that glycosylation is not done by the virus, it's done by the host machinery,
Host well. Hmm.
right? So if
Yes,
you target glycosylation, you are going to target also the
the
host
host
machinery
well.
that's
Yeah. Yes, sir. Yes, sir. I got the point, sir. So, do you think if, for God's grace, if we develop a vaccine which is effective, highly effective for all the strains of the virus, do you think different people have a different biological response towards it? Because everybody is not physically healthy. Somebody will have diabetes, somebody will have some other complications, some respiratory disorders. So, do you think that the bodily response will also affect the vaccine which is being administered to the patient?
see, inter-individual variability in response, whether it's to treatment, whether it's to a vaccine, it's very well
Mm.
documented. And this could even be an infection, right? Not all people respond the same to an infection. And this is inter-individual variability. And this inter-individual variability could be caused by a multiple number of factors. This could be environmental factors. This could be the dosage of the drug, therapeutic, vaccine, or infection. This could be the bioavailability or the pharmacokinetics and the pharmacodynamics of that particular molecule. This could be influenced by, in some ways, the genetics of the host. Now, there are very many factors which could influence inter-individual variability. And this inter-individual variability is all across. You take any
Mm.
therapeutic, right? You
Mm.
take any drug, you take any vaccine, you not all people respond the same
Same way.
way that you expect, right? So this is very well documented and is very well known, right? All
Mm.
people don't respond the same way. Now there are very many factors which could contribute to how people respond very differently. And what's really important is to understand each of these factors, or at least the major factors which could cause their individual differences. For some molecules, we know these are genetic causes. Some
Yeah.
some molecules we know these are environmental causes.
Mm.
Some molecules, we know the general metabolism of that particular drug or the molecule is different in different individuals. This could be, for example, the gut microbiota. This could be the environment in which the individual lives. This environment means what are the other food that the particular individual takes, so on and so forth. So it's very important to
Yeah.
understand it. Or other words, what I would suggest is, what we really need to do is to do more good science to be able to understand this much better, rather than now trying to build translation on poor scientific basis. So we need to first understand the basis in much greater detail, and once we understand that, of course, that would have a lot of value in translation.
Okay. Okay, sir. Okay. Thank you so much, sir. Now we'll move towards the questions that been asked by the audience in the chat section. I would request you, sir, to kindly sort some of them out. Um, the first question is from in how much time we can expect a variant characterization, whether the variants are a result of adaptation to the host system or the result of the resistance to medication.
Okay, so in coronavirus, the mutations that occur are supposed to be occurring just by random,
Okay,
right?
sir.
Now, having said that, the origin of the mutation can be random, but the evolution of the mutation could not be random. The best example that you could say, or you could sort of hypothesize, is that if you have a virus that is mutated,
Hmm,
right, which makes
hmm.
it dysfunctional. For example, it lost the RDRP protein,
Okay.
right? The occurrence of the mutation is by a random event,
Yeah.
right? But now that the virus doesn't have RDRP, it cannot
Hmm,
reproduce further,
hmm.
right? That means that going forward, you will never find a lot of viruses with RDRP mutations, because the RDRP mutation containing viruses have all died,
Yes.
right? So
Yeah.
what you see at this point of time are snapshots. So you need to
Hmm.
take snapshots over time in a temporal fashion to be able to understand what came in, what happened, and what evolved and what emerged out. And that will allow
Okay.
us to understand what caused these viruses to emerge. Is it because they could infect better? Is it because
Hmm.
they could replicate faster? Right? So on and so forth.
Yeah. Okay, sir.
Right?
Okay. Okay, sir. Another question is sir, the vaccine which are developed in India, are they developed from the strains of CoV-2 which come to India from, or also from the strains from worldwide?
Frankly speaking, I have no idea, because I don't see any
Yeah.
literature on the vaccines or the strains which are available. So I would not
Okay,
like
sir.
to comment on that.
Okay. No problem, sir. Okay. So the next question is that people who get infected with CoV-2 show varying symptoms. Some show very severe symptoms while some are asymptotic. Oh, sorry, asymptomatic. Sorry to. Do you think SNPs play a major role in this?
Yeah, so that is what I said earlier, that this is essentially an example of inter-individual variability in the
Yeah.
disease manifestation,
Mm,
right? And now, inter-individual variability essentially means that a large number of people, which is also documented, are asymptomatic or with much less number of symptoms, so much less
yeah.
severe symptoms. A
Mm,
very small proportion of individuals have more severe symptoms,
mm,
right? Now, globally, what is known is that people who have a higher age,
mm,
people who have comorbidities,
mm,
have a higher incidence of developing much severe disease,
mm.
right? Now, having said that, it doesn't mean that these are the only factors,
Yeah.
right? There are additional factors which have come up very recently, like, for example, smoking, which is an
Yeah.
environmental factor,
Mm-hmm.
which then, of course, you could have race, right? The
Yeah.
blacks, Asians versus Americans or the Caucasians
Mm,
have very different rates of developing severe infections.
yeah.
So, there are very many factors, right, which
Yeah.
can change the symptoms or the severity of the disease. Now, no one individual is going to have one factor only,
Hmm.
right? It's a combination of many factors which come together, which can make the disease severe or less severe. Now,
Yes.
having said that, at this moment in time, we have only observational values. That means that we have looked at people who have less severe symptoms. We have looked at people who have more severe symptoms, and
Hmm.
asked the question: Are you a male or a female? Are you black or a Caucasian or an Asian? Right? Do you
Hmm.
have hypertension? You don't have hypertension. Do you smoke? Do you not have smoke? So these are observational studies,
Yes.
right? Now, what we need
Yes.
to build is from these observational studies biological plausibility, right?
Yeah.
For example, answering this question, why do smokers have more or less severe symptoms? So there should be some
Yeah.
biological plausibility or biological mechanism of
Yes,
explaining
sir.
this. So
Definitely, sir.
until we have a biological mechanism, these are only statistically significant observations,
Yeah.
right?
Okay. And sir, I would like to also add one thing to it, that I have observed that people suffering from respiratory disorders, they have shown more severe and fatal infection due to coronavirus. People having some other disorders like hypertension or any cardiovascular disorder, they are not much infected. I don't know how far I'm correct. This is what I read somewhere, but people having respiratory disorders have been affected more. Do
I'm not
do you
really
agree?
sure about that observation. I'm really not
Mm-hmm.
sure about that observation. But of course,
Okay.
what you can read through is there is a very recent observational study which has come from US on a very large number of comorbidities across a very large number of samples. I
Mm-hmm.
think it has appeared just yesterday or day before yesterday. I could do one thing. I could post the link on the chat in a few minutes.
Yeah,
So this
sure, sir.
is probably one of the most exhaustive studies done.
Okay, sir. Sir, the next question was how scientific community can equip themselves for keeping a surveillance for the new emerging virus, the one we discussed regarding the diagnostic kit, failure of diagnostic kit. So, as you said that it might fail. So, regarding that, there is this question that how scientific community can equip themselves.
Yeah, so it's a very interesting question because yeah, uh, there are few things that we learned by going through the epidemic, right?
Hmm.
Because in, at least in our lifetimes, we have not seen another epidemic of these large proportions,
Yeah,
right? So there are of course things that we learn from this epidemic, and that's
yeah,
in retrospect.
yeah.
Uh, and of course, there are things that intuitively come to our mind
Hmm.
which are not essentially learnings, but this is how humans react to a new sort of an epidemic. And you would see this very often in real life situations. But having said that, the most important thing going forward, or in terms of the learning that we have, is one: uh, can we identify the pathogen much earlier than
Hmm.
previously thought? I mean,
Hmm.
can we identify pathogen in, for example, a few weeks or a few days through the epidemic or through outbreak?
Hmm.
Can we use that information in terms of the genomic information to quickly develop diagnostic reagents,
Yeah.
right? And can we follow up this epidemic over time in
Hmm.
understanding, are the diagnostic reagents failing, or can we
Yeah.
build new diagnostic reagents? So I think that is what is really going to change most epidemics going forward, because
Hmm.
as long as we can diagnose patients faster, efficiently, we can avoid
Yeah.
a lot of deaths which are avoidable,
Okay,
right?
sir.
But but there are a lot of other things that also need to come in place, which includes better understanding of how the virus acts, how the
Hmm.
virus causes a disease, how the virus transmits. And this will be useful to build new ways of controlling the virus, treating the virus, new ways of developing new vaccines,
Hmm.
right? So there's a lot of new things that we are learning, that I think is going to be impactful in the future epidemics that come. And epidemics are
Yes.
going to stay
Hmm.
with humans,
Hmm.
right?
Okay, sir. Sir, there was one more question, that is there any synergy between variations in virus or host and susceptibility?
At least there is one very recent paper which appeared in Cell, which suggests that the D614G mutation, which is in the spike protein, can change the infectivity of the virus. So, to give a bit brief background about this particular genetic
mm,
variant, this is a variant which essentially is one of the variants which classifies the A2A clade, and A2A
yeah.
clade is globally the largest clade which has emerged
Mm,
across the world and also is emerging in the country.
mm,
So what they have shown is that this particular genetic mutation, the spike protein, allows it to be more infectious, and then therefore it's an advantage to the virus to emerge faster. So answer is yes. In terms of the
okay.
host genetic markers, yes,
Mm,
there are a lot of markers that people have looked at, and people have actually there's an ongoing host genomics initiative, which is a global
mm,
initiative to look at host genetic factors.
mm,
In many ways, we are far from finding a good biomarker which can. A good biomarker means with a very good odds ratio which can modulate the host response. Of course, there are many more studies on rarer variants which
mm,
are impactful in smaller number of patients, especially for people with rare genetic disorders.
mm,
There are a lot of papers, for example, people with immunodeficiency, primary immunodeficiency, so on and so forth. But that's, of course, having said that,
yeah,
they are host genetic factors, but
yeah.
they're not really going to be applicable to a wider community.
Okay, sir. Okay, so one more last question is there. Do you think we should sequence genomes of coronavirus patients? Would this now play a role in evolution given the integration of viral genome in patients?
Yeah, so we should. So my answer to that is we should sequence coronavirus patients, not all patients, but very specific subgroups of patients
Hmm.
uh, who deviate from the expectation.
Okay.
Now, when I say expectation, of course, I'll come to back in a few minutes.
Hmm.
But just sequencing any patient with coronavirus probably is not going to be that useful,
Hmm.
um, just because of the fact that it's impossible to just sequence millions of people,
Yeah.
right?
Yeah. It is. Yeah.
But yeah, so to answer that question, yes, we need to sequence individuals. We need to sequence individuals who deviate from what we expect, right? When I say
Okay.
what we expect is that, for example, if a patient is, for example, young, right?
Hmm.
Uh, mm, Given the profile of the disease, most young people are supposed to have much lesser severity
Hmm.
of infections, or in many ways no symptoms at all. But if, for example, a young individual with no apparent comorbidities, no apparent health conditions before,
Huh.
develops a very severe infection, right?
Hmm.
That is departure from the expectation, and then therefore that is important to sequence and figure out what is really different in this individual. And
Yes,
the
sir.
same on the other extreme of observations, like, for example, if you have an old individual, and from press I see a set of old individuals who have seen the previous epidemic of influenza. That means
Hmm.
they're more than 100 years of age,
Hmm.
right? And if they have survived this epidemic also, right,
Hmm.
and they're still healthy, it would be really interesting to note what is the genomic component.
So there have been such cases in America where there was this lady who was approximately 115 years old. I exactly don't remember the age. She had overcome the last pandemic. She got infected this time and she
Good.
recovered. And she
That's right.
has not till date
So they are,
got it again for the
yeah.
second. Yeah.
So they are individuals in India also who have seen the previous pandemic and have
Hmm.
survived this pandemic also, and
Yeah.
they would be very good candidates to go back and sequence.
Yes, sir. Because there is also I don't know whether it's a myth or not, that people of old age or the senior citizens are more susceptible and it can be more fatal for them. But there are individuals who have recovered even after getting infected at such age.
Absolutely. And to add to that point, there are also a very large proportion of patients,
Hmm,
especially patients with genetic disorders,
hmm,
uh, typically patients with immunodeficiency, for example, primary
hmm,
immunodeficiency.
hmm,
They have either any one of the arms of the immune system which is lost. For example, in
hmm,
extinct gamma globulinemia, you would have the
hmm,
B cell or the immunoglobulins absent. So such
Hmm.
individuals would respond very differently to COVID,
Yeah.
right? And
Yeah.
if they respond very different to COVID, it would be really worthwhile to sequence them and understand why they respond very differently to COVID. Because
Yeah. Yes.
the expectation in primary immunodeficiency is that they will not survive COVID
Hmm.
because they do not have
Exactly.
uh, a patent
A
immune
strong
system.
immune system.
Yes, but literature says that at least there are 13 cases which has been described in literature with people
Hmm.
with primary immunodeficiency disorders and their outcomes in COVID. The observation is not matching the expectation, and then therefore you
Hmm.
should go back and look at the genomes to understand, since these are also genetic disorders, to go back and
Hmm.
understand why they respond very differently.
Yeah. Yes, sir. So, we now come to the end of our first webinar. Thank you so much, sir, for taking out time from your busy schedule and collaborating with us. I'm sure that the audience has gained knowledge and put rest to the few of their queries regarding coronavirus. I would like to thank the scientists who have graced our first webinar through their presence. We would also like to thank our enthralled listeners for cooperating with us. We are doing this for the sake of knowledge and to spread the awareness. Your presence for our webinar is highly encourageous and supporting for us. We will be back with our next webinar, which is going to be held on 12th July 2020, that is this Sunday, 5 p.m. onwards. The guest speaker is Dr. Arindam Mehta from National Institute of Biomedical Genomics, Kalyani. We will discuss some more points like how do the data and various analysis tools help in the detection of viral genomes, how bioinformatics is helpful in cooperating in distinguishing different strains of the viral virus, what role can genomics play in curbing the second wave, and so on and so forth. Looking forward to see you with the same enthusiasm and zest. For further updates, stay tuned to our website, YouTube channel, and social media handles. Thank you so much and have a great day. So
Thank you
your
very
mic.
much.
Thank
Yeah,
you so
thank you
much.
very much.